Okay, lesson 24, synaptic transmission. In this lecture, we are going to cover the events involved in synaptic conduction. How are the neurotransmitters categorized? The effect of neurotransmitters on membrane potential, and the mechanism to remove those neurotransmitters from synaptic cleft. What is synapse? The structure that neurons use to communicate with other neurons. So if you take a look at this picture, you will see that one neuron is connected to the other, and there are billions of those neurons which are actually communicating with each other. That junction, another word for synapse is junction, and this is the neuronal synapse. There are three types of synapses: axodendritic, axonic, and axosomatic. First type is here. You see that circle area right over there. Uh, the axon of one neuron is synapsing with the dendrite of another neuron. So one axon, another dendrite. So this is the synapse called axodendritic. In axosomatic, soma is body. So the connection is between the one axon and the body of another neuron. So axosomatic. Third one is axonic. This time synapses between one axon and another axon of another neuron. So that is will be the third one. So axodendritic, axosomatic, and axonic. Three types of synapses. Now, pre and post synaptic neurons. Just to simplify, look at that blue area. The presynaptic neuron sends message from its axon. That's the axon terminal. So this is the presynaptic. Pre means before. The pink area, that's the postsynaptic neuron, receives message from its dendrite, cell body, or axon. So pre versus postsynaptic. In previous lectures, we discussed about the synapse between nerve and muscle, and I'm sure you can recall that. It was called neuromuscular junction. Now, what is the difference between a neuromuscular junction and a neuronal synapse? Here you go. In a neuromuscular junction, we found that muscle fiber receives input from a single axon. One single axon is sending that input to the muscle. But in neuronal, the neurons may receive input from thousands of axons. It's much more complicated. In NMJ, neuromuscular junction, only acetylcholine excites these muscle fibers. That's the only receptor, only neurotransmitter, acetylcholine. But in neuronal synapses, there are several of them. It may excite or inhibit the postsynaptic neuron. In NMJ, action potential in neuron triggers an action potential in muscle fiber. Whereas in neuronal synapses, a postsynaptic neuron may require input of 100 or more presynaptic neurons. Transfer of chemical or electrical signals between neurons at a synapse is called synaptic transmission. It can be electrical or chemical. Think about the breathing process, which is a programmed behavior, right? Every minute, 12 to 20 times, our muscles are breathing. They are working through some neuronal impulses are there. And this type of synapses are electrical synapses. They have very small separation gap, 3.5 nanometer. And the gap junctions you see here in this picture allow the electrical current to flow from one neuron to the next. Okay, so very small gap, and there are gap junctions as well. And these transmissions are bidirectional, that means they can move back and forth, and it happens nearly instantaneously, zero point 0.1 millisecond, 1/10 of a millisecond. You think about it, so tiny fraction of a second. So for the electrical synapses, these are the characteristics. Let's take a look at chemical synapses. Majority of the synapses in nervous system are chemical synapses. This time they are unidirectional. That means one way, unlike the bidirectional in the electrical one. And these electrical signals converted into chemical signals with no loss of strength, and it is slower with 0.5 millisecond. Remember, the other one was zero point 0.1 millisecond. Now it is 5 times slower. An advantage is that it can be controlled, it's not programmed. Presynaptic neurons have synaptic vesicles with neurotransmitters inside, and the cleft is 20 to 50 nanometer. So there's a gap between pre and postsynaptic neurons. Postsynaptic neurons have receptors for neurotransmitters. Receptors usually linked directly or indirectly to ion channels. Signals can vary in size. Now let's take a look at those events at this chemical synapse. There are 4 events taking place. Number 1 is the action potential. In the presynaptic neuron, when the action potential arrives, it will trigger the calcium channels in the axon terminal. Okay, that is the first thing to happen. Action potential in the presynaptic neuron triggers calcium channels in that area. And then step 2, when the calcium channels are open, there will be influx of calcium, causing synaptic vesicles to release neurotransmitters into the synaptic cleft. Step 3, the neurotransmitters will bind to receptors on the postsynaptic neuron. And then the ion channels will open, leading to local potential and possibly an action potential. So these are the 4 events that happen in chemical synapse. Just to summarize, first, the action potential will make the calcium channel to open. Calcium influx causing the vesicles to release neurotransmitters. Neurotransmitters will bind to the receptors of the postsynaptic neuron, and then ion channels open, leading to local potential and possibly action potential. Now, there are 3 ways to stop this synaptic transmission. So response can be stimulated again, because we have to stop this thing. It cannot go forever. What are those 3 ways? Number 1 is the diffusion and absorption of neurotransmitter. You see on the left side of the picture, those red balls, neurotransmitters, they are diffused. I mean, they are spreading around and they are absorbed in the neurotransmitters. Those things are absorbed and diffused. That's one way to remove them. Second thing is the degradation. There can be some enzymes, and in that area, which will degrade these neurotransmitters. So degradation in the synaptic cleft. Thirdly, that will be this reuptake into presynaptic neuron. So they are taking it back from where they came from, right? So 3 things. 1 is the diffusion and absorption. Second, degradation. And third is reuptake. This is how the neurotransmitters will stop its function so that there will be another impulse work on the same synapse. These receptors, the neurotransmitter receptors, can be of two types, ionotropic and metabotropic. This one is ionotropic. Receptors that are part of the ligand-gated ion channel, and they are rapid but short-lived effect. You see here in this picture, uh, this channel is open, is closed at first. This is the ionotropic receptor. And when the neurotransmitter bound to the receptor side, that red dot you see here, it will open the gate so that ions can move. This is ionotropic receptor. In the second type, we have metabotropic. This time, the receptors interact with a separate ion channel, not the same one, like the previous. We discussed that one, that worked on the same place. But here, you will see that it works on a separate ion channel, and they mostly interact through the intercellular enzyme called G proteins that forms a second messenger to open or close the channel. It's a long lasting and variable, and more variable. Take a look into this picture. What do we see? You see that on the left side, this is the metabotropic receptor, that orange color protein, and it is connected to a G protein inside the cell, in the cytosol. And the blue gate, the ion channel gate, is closed now. The moment neurotransmitter, that red dot, that is square, it is bound to the receptor, this will activate the G protein, that green color. G protein is activated. There will be enzymatic reactions. It will make second messenger. That second messenger will make this ion channel to open for the ions to move in and out. So this is metabotropic receptors. Just to simplify and to know what is the difference, in ionotropic, it opens the same channel where the ligand is attached. In metabotropic, it's a different channel they will be opening. Major neurotransmitters can be classified into excitatory and inhibitory and neurotransmitters. So if it is excitatory, it will cause EPSP, which is excitatory postsynaptic potential. If it is IPSP, inhibitory postsynaptic potential, that will cause this inhibition in the postsynaptic potential. This will be much more discussed in other lectures, but for now, just remember EPSP and IPSP. Now, the last part of this short lecture is about the type of neurotransmitters. There are 4 groups of neurotransmitters. One of them you are familiar with is acetylcholine, which was discussed in neuromuscular junction. But in nervous system, this is one of the 4 group, acetylcholine. There are many more monoamines, amino acids, and neuropeptides. Acetylcholine in nervous system, it is found in cholinergic synapses. So cholinergic, acetylcholine, right? You can correlate those 2 words. And they are found in neuromuscular junction, brain, spinal cord, and autonomic nervous system. Mostly excitatory, stimulating the next nerve. And they are degraded by this enzyme acetylcholinesterase into acetic acid and choline. So this enzyme plays an important role for degradation of acetylcholine. Second group, the biogenic amines or monoamines. Examples are catecholamines like norepinephrine, epinephrine, dopamine, those words by adrenaline, noradrenaline. Sorry. Serotonin, this one is involved in mood regulation. Histamine has diverse functions. In the third group, we have amino acid neurotransmitters like glutamate. glycine, or gamma aminobutyric acid, okay, GABA, that one. In the fourth and last group, these are the neuropeptides, substance P, which functions in pain and temperature, opioids, that includes endorphin, enkephalins, dynorphins. They are also for pain relief and depressants. And feeding behavior, our feeding behavior involves this neuropeptide Y. So there are many more, hundreds of them are so far discovered. But for this course, we just need to know a few of these names in four groups. Once again, there are four groups of neurotransmitter: acetylcholine, biogenic amines, that is monoamines, amino acids, and neuropeptides. So that is all for this lesson.